Hello Year 11, this is Mrs Jones and I'm going to talk to you today about genetic engineering. So today's learning objective is to know how and why an organism is genetically engineered. As always in the lesson we start with retrieval practice, so pause this screencast now and please answer the 10 questions on this slide. There are 5 from biology from year 10 and 5 from biology this year. Okay, hopefully you paused the screencast and answered the 10 questions on the previous slide. So the answers are there. If you have got a green pen, then use the green pen to self-assess. If not, just use whichever pen you've got to self-assess your 10 answers. If you did get any wrong, please make sure that you write down the correct answers. It will help you in the future remember them. Okay, so today's lesson. We are going to find out how and why an organism is genetically engineered. So there's your definition. I would write that down into your book or onto a piece of paper. Genetic engineering is when a scientist alters an organism's genome to produce an organism with desirable characteristics. Now, if you look in the title of that slide there, the gene and the engineer are in bold. Okay, that's essentially what genetic engineering is. It's manipulating the genes of an organism to have desirable characteristics. So there's a couple of pictures on there. So you could make certain vegetables have more nutrients, or they could engineer a leaf to be frost resistant, or they could engineer a plant to be resistant to a pest. So all of those things are beneficial to the plant and therefore useful for a farmer or for whoever is growing the crops. Okay, so moving on our next slide, here are some reasons why farmers would want to genetically engineer their crops. So you might want them to be stronger or taller, increase yield. So that yield means the amount of product that they have got. And the farmers may want them to be freeze resistant, pest resistant, or to produce toxins to kill pests. Now, all those things actually enable a farmer to have more crops. And what does more crops mean? More crops means more money, okay? Which actually is what the farmers need. Although they often are so passionate about their job, they inevitably need to make money. So all these things will help them to make more money, and it means that farming will be sustainable. Okay, please just pause the video and jot those 6 ideas down underneath the side heading, why might farmers want to genetically engineer their crops? Okay, moving on then, I just wanted to share with you some examples of genetic engineering. So if you see that first image there, that would be cotton that has been genetically engineered to increase the amount of cotton that is actually on those plants. Again, more cotton, more money. The next one would be corn, and that corn there has been genetically engineered to produce toxins that actually kill insects. Those insects that normally kill the corn will die if they try and eat the corn, which again protects the corn, enables the farmer to make more money. Now, you may not know much about genetic engineering, but you probably have heard of GM foods. So GM foods are often all over the news. You can read about them in scientific magazines. And GM just means genetically modified. So if you hear about a GM food on the internet or you read about it, the GM food is something that has been genetically engineered. So the two things directly interlink. Now, the last example there is the use of bacteria. Now, bacteria asexually reproduce. That means that they do not have to have another bacteria to help them reproduce. They just make copies of themselves. Now, because bacteria reproduce at such a ridiculously fast rate, they can be used really usefully in making drugs such as insulin. Now, insulin, as you should know, is used to lower blood glucose levels. Years ago, we actually took insulin from pigs' pancreas. Now, some people may have been against that because they were vegetarian or vegan, and it isn't the best way of doing it. So what scientists have realised is that we can actually manipulate bacteria, genetically engineer them to make insulin. Now, they don't naturally produce insulin, but we can give them a gene, genetic engineering, to enable those bacteria to release insulin, which the scientists can collect and then they can use to help people who are diabetic, which is an incredible use of genetic engineering. I'm sure you'll agree. Okay, I have sent this for you to use, but if not, you could just take a little sketch of it by pausing the screencast and drawing this process. This is a process that you do need to know the stages for, so it might be short answer questions, there might be a longer answer question in the exam, which could be up to 6 marks. So I'm going to talk you through it, the steps involved, and if you have got a copy of it or if you have drawn it, please write each step down. So, step number 1, the genes which code for the desired characteristic, and in this instance we're going to talk about the production of insulin, they are actually cut out of the genes of a human cell. So scientists get a human cell, they get the genetic information and they know which gene codes for the production of insulin. Insulin is a protein, by the way. They remove it using restriction enzymes. So those little orange triangles on the slide in front of you are restriction enzymes, and they cut out that foreign gene which enables whichever cell it's in to make insulin. So we have now got the gene that tells whatever it's in to make insulin. Okay? Step 2 at the bottom. So bacteria, mentioned this before, they multiply really quickly. So what the scientists do is they remove the plasmid, which is a ring of DNA. They actually cut the plasmid, again using restriction enzymes, so please label then if you have drawn the diagram. And hopefully you can see at stage 3 that the foreign gene, which codes for the production of insulin, is put into the plasmid. So the plasmid now has got a gene that codes for the production of insulin. That plasmid is put back into a bacteria, and stage 4 is that asexual reproduction of bacteria. So every bacteria that is made of the asexual reproduction, so one bacteria quickly becomes 2, 4, 8, 16, 32, 64, 128, 256, 512, 1024, every single one of those bacteria that are made through asexual reproduction will actually have the ability to make insulin. So you have now got thousands of bacteria that release insulin. The scientists can then collect the insulin and it can then be distributed to pharmacies, and obviously people who are diabetic will be able to collect it and use that insulin. Okay, so I'm just going to ask you 5 questions now about this process. So if you could just jot 1 to 5 down in your books, please. Okay, question number 1. What is the enzyme called that is used to cut the DNA? What is the enzyme called that is used to cut the DNA? Question number 2. Is this process faster or slower than selective breeding? Question number 3. Which part of the bacteria is removed so that the foreign gene can be inserted? Question number 4. How do bacteria reproduce? Is it asexually or sexually? Question number 5. Are all the bacteria similar, the same, or different when they reproduce? Okay, well done if you've had a go at those questions. Here are your answers. So number 1, the enzyme is called a restriction enzyme. Number 2, genetic engineering, you are changing the genome of the organism, therefore it is faster. It happens immediately. Number 3, the part of the bacteria is called a plasmid. Number 4, bacteria reproduce asexually. And number 5, they make exact copies of themselves, which means that they all have that foreign gene in, whatever that foreign gene may be. Okay, so it isn't always bacteria that are genetically engineered. Some other organisms can also be genetically engineered. So it isn't just about a bacteria having the genes manipulated. There may be manipulation of genes of other organisms. So if you have a look at this question here, describe the steps to genetically engineer tomatoes to become frost resistant. The flounder fish can survive in very cold waters. So I do want you to have a little go at this yourself, but the idea is, first of all, work out which organism has to be genetically engineered, which is the tomato, and also which organism has the gene that is useful. So in this case, it is the flounder fish. So just to give you a little bit of a starting point, the flounder fish has got the gene in that is needed. The tomato is the one that the farmers, the scientists, want to make frost resistant. So see if you can use the content and the concepts from the previous slide to work out what is going to happen. Look at the pictures on this slide and have a go at writing down the stages of genetically engineering this tomato to be frost resistant. If you would like to send your steps to me so I can have a look and mark it, that's absolutely brilliant. If not, you can show me on your return to school, but have a go and try your best. Okay, so the last thing now, which is really important, is this comparison of selective breeding to genetic engineering. Now, this is often a comparison question in exams, 6-marker. This idea of selective breeding versus genetic engineering. So if you've got a book or a piece of paper, please get this table jotted down. So you've got genetic engineering, which is quick because you are changing the genes and you get the desired characteristics immediately. Okay. Disadvantages of genetic engineering is that people think it is playing God. So you will find lots of articles online about genetic engineering and people not being happy with GM crops. We also, because we've not had genetic engineering and GM crops for a long time, we've no idea of the long-term impact on other plants or animals in the food chain. And some people just think it's unethical. Okay. Now, on the flip side, we've got selective breeding. People say we've done this for thousands of years, so why should we stop now? But again, we can get the desired characteristics, so definitely advantages. For just selective breeding, because it is inbreeding, so you are getting animals from the same family breeding, the gene pool will get smaller, which will mean the harmful recessive alleles will build up, which can lead to health problems. And people do say that this, of course, is cruel because the animals do end up with health problems. Okay, right. I'm going to leave this screencast here. So please, rewind it if there's anything you'd like to go over again. Pause it, go over the questions. You will also have some exam questions attached to this email that you can have a go at, which you can do following this work on genetic engineering and following the work on selective breeding. Have a really good day and work hard. Thank you.